Is the screen visible to all of you? Yes or no? Fatima, Arpit, Samaira?
Yes, sir.
Yeah. Okay, so where were we in the last class? We were on law of independent assortment. Okay, so quickly, can anyone just give me a quick recap of the 3 laws? Fatima, how about you? Just the 3 laws of Mendel. Or let's start from exceptions of the law of dominance. Yeah. This is what we started from. Fatima.
Sir, can
Would you
someone
like?
do it?
Sorry?
Can someone else do it?
Yes, someone else is Arpit and Samaira. So who would like to take it for
last class.
Okay, then Samaira.
Yes, sir. So
Yes.
the exceptions of law of dominance, and the first one is incomplete dominance.
Mhm. Yes, continue.
So we took the flower color of snapdragon. It has 2 colors, red color and white color. Then we crossed both. Then we got a intermediate, that is capital R and smaller, that is pink flower.
Yes.
Then we self-cross the F1 generation, that is capital R and small r. Then we got the
Yes.
phenotypic ratio as 1:2:1 and genotypic ratio also as 1:2:1.
Yes, right. So which means that incomplete dominance affects the phenotype, but not the genotype, right? The genotype is same as it was.
Yes. So incomplete dominance is a type of gene interaction in which both the alleles of the gene are partially expressed, and in a heterozygous condition, no one allele is able to exert dominance over the other.
Yes.
This results in intermediate or different phenotypes. And the second exception is co-dominance. It is a type of gene.
Yes, continue, please.
This is a type of gene interaction where 2 alleles of the same gene in a heterozygous condition are expressed separately. And equally to give rise to different traits in same organism. We took the example of AB blood group. It has both A and B antigens are produced on the AB blood group. Has 2 antigens, that is A and B. Then
Yes.
the second law is the law of segregation. It is also known as the law of purity of gametes. It is a universally accepted law, and it is defined as it states that when an organism makes gametes, each gamete receives a single copy of allele for each gene, and also it can also be defined as it states that alleles are independently segregated to form alleles. And the third law is law of independent assortment. We did a dihybrid cross with we took the character of plant as height and seed shape. Then we crossed 2 plants, one plant with a tall with a round seed, and the other is dwarf plant with wrinkle seeds. Then we got the FN generation as capital T small t and capital R small r, that is hybrid tall and hybrid round. Then we self-cross the FN generation. Then we got the genotypic ratio as 1:2:1:2:4:2:1:2:1, and the phenotypic ratio as 9:3:3:1.
Yes, right. And then I also told you some handy formulas. One of the formula is this that you can use. Remember this formula to find out number of gametes of any genotype, right? 2⁻n, where n is the number of heterozygous genes or alleles. Remember? Fatima found it very handy and useful. And apart from that, there was another one to find the total number of phenotypes, which was this one. How many combinations you will get? Sorry, the total number of genotypic combinations that you will get, 4⁻², which is the number of genes. If we take a dihybrid cross, it is 2. If we take a trihybrid cross, it will be 3. So these two formulas, just remember, it's very important. I think I have not told you what exactly is law of independent assortment, right? Have I dictated you that? I think we did the dihybrid cross and then stopped there.
Yes, sir.
Yeah. Okay. So write down the law of independent assortment states. Law of independent assortment states that the alleles of 2 or more different genes the alleles of 2 or more different genes get sorted into gametes independently of each other. Independently of each other. That is one gene does not influence alleles of one gene does not influence the alleles of another gene. So it is like the law of segregation, but it's not for the different alleles of the same gene. It is given for two or more different genes. Do you understand and appreciate the difference, everyone? So the definition or the statement for law of independent assortment is very, very similar to the law of segregation, right?
Yes, sir.
Yes or no? But law of segregation talks about just the alleles of one single gene, which means it does not talks about one gene's interaction with another gene. Okay, it only talks about the alleles of one gene segregate independently to make gametes, and that's always true. That always happens. Okay. But when it comes to alleles of 2 or more different genes, where 2 or more different genes are present together, then this law, the third law, law of independent assortment, states that one gene does not influence another gene. Okay. And the alleles of both the genes are independently segregated from each other. Now, this law was holding true, but unless there was a scientist whose name was Morgan, who came and figured out. So the kind of experiment that Mendel did on pea plant. So look at this dihybrid cross here. He took two characters, tall and round, right? Height and the seed shape. Is it clear, everyone? And he saw that all different combinations were possible: tall with round, tall with wrinkled, dwarf with round, and dwarf with wrinkle, right? Everything was possible, and this can only happen, and the ratio was 9:3:3:1. Now in this ratio, pay attention, this 9 is parental, right? This 9 is what? Tall and round. Am I correct? And this one is dwarf and wrinkled. Am I correct?
So,
Yes, sir.
Now these two, 9 and 1, are only parental types. The parents were either tall or round when we started with. Both the parents were tall and round here, and above also either they were tall and round or dwarf and wrinkled. No parent was dwarf and round and tall and wrinkled, right? So these two, 3:3, these 6 combinations out of 16 were recombinants. Do you understand? So according to this ratio here, 6 out of 16 combinations were recombinants. Do you understand? Say yes or no.
Yes, sir.
Now, can anyone tell me what is in percentage, what is 6 of 16? How much percentage is 6 of 16?
sir, what did you ask?
6, what percentage is 6 of 16? If you got 6 marks out of 16 marks total, what will be your percentage?
That's 75%.
What?
That's
Not at
70.
all. 6 marks out of 16 cannot be 75% because the half of 16 is 8. So 8 will be 50%. How are you calculating? If you received 8 marks and the total was 16, you got 50% marks. And if you got 6
37%.
marks, how much?
37.5.
Are you sure?
I guess.
Yes, it's around 37 point something. Let's say it's 37%. So 37% of progenies are what? Recombinants, right? Different from both the parents. Yes or no? This is how variation comes in sexual reproduction. Do you understand? If you follow a normal rule where you are studying 2 genes, then 37% of the progenies, somewhere more than 1/3, will be recombinants, not like any of the parents. Now, this was true for pea plants, and Mendel thought that it will hold true for every other organism. Unless and until Morgan came and did the same experiments. He started looking at 2 different characters, but in a different organism known as fruit fly, Drosophila. Okay, where he figured out for the first time that it's not true for all the genes. So let's come to exceptions of law of independent assortment. Because I have already told you, it is not a universally accepted law, correct? Yes or no? People?
Yes,
It is your
sir.
energy. Yes. So if any dihybrid cross follows this ratio, like this percentage of recombinants, which means it is following Mendel's rule. But when Morgan, so write down the exception to this law is known as linkage between genes. Linkage between genes. Now, what does the word linkage means? Simply
Connection.
physical connection, right? That is what is linkage. Now, the scientist called Morgan. Okay. So first, let's define linkage of genes. Write down. Linkage between genes. Linkage between genes is like functional association. Is like functional association. between two genes that does not allow it to segregate independently of each other. Is it clear? Now, giving an example of this, let's say Samira and Fatima are best friends. Okay, so whenever and wherever they go together. So if there is a party, and if you find Fatima in the party, and you know that they are best of friends and the linkage between these two is 100%, then you know that you will find Samira also there, right? Yes or no?
Yes, sir.
And if it is 50% of linkage, then the chances will be 50% that Samira may or may not be present there. And if Samira and Fatima are just not friends at all, like they just know each other, but they are not friends, so there is no chance, you can say that anything can happen. So linkage and recombination has a inversely proportional relationship. More the linkage, less the recombination. Okay, you understand? And less the linkage between two genes, more the recombination. Is it clear, everyone? This is the formula. Now, how it was found? So write down. Linkage was discovered by scientist called T.H. Morgan, Thomas Hunt Morgan. T.H. Morgan discovered the linkage between two genes in Drosophila. Okay, Drosophila is fruit fly. It's a small fly which is attracted to fruit odor. Okay. Now what he did was, he did similar thing that Mendel was doing with pea plant. So in Drosophila, he took a yellow bodied and white eye fly. Yellow bodied, white eyed female, let's say. So yellow body is the gene for body color, right? Now Drosophila do not have skin, but they have the body. So their body was yellow and their eyes were white. So these two are different genes, right? Giving rise to different characters, just like tall plant with wrinkled or round seed. Yes or no?
Yes.
Now when he crossed this with brown bodied and red eyed male. Now normally you expect that this is another dihybrid cross, and let's say if we write the genotypes, then it will be YY, WW. This will be small y, small w. So you will just expect that this is just another dihybrid cross, and in the F2 generation you will find 9:3:3:1 phenotypic ratio, right? So most of the flies will be yellow and white, and then one will be brown and red, and 6 will be the combination, which means 37% will be the recombinants. Yes or no? Which is yellow and red and brown and white. This is also possible, right? But this did not happen here because this yellow bodied gene was actually linked to this white eyed gene. So there was linkage between these two genes. Okay, so these two characters, these two genes showed linkage. Now, if they show linkage, how will we figure out in the next generation? The ratio will differ. So when you will cross them, you will make F1 and then you will self cross F1. Then he saw that the phenotypic ratio, did not follow 9:3:3:1. So this was not followed. And what does this mean? This means that these recombinants were not there. You understand? You understand, people? Am I clear, everyone?
Yes, sir.
So he found the number of recombinants. Recombinants are non-parental type. Non-parental combination or types. The number of recombinants were very few. And most of the flies were either yellow and white or brown and red. Okay, which means whatever was the parental genotype, it just got inherited the same in the next generation, right? Which means these two genes could not independently get segregated. So the law of independent assortment between two genes did not work here. Is it clear, everyone?
Yes, sir.
Yeah, very good. Now, before going into recombination and other things, let me also tell you about chromosomal theory of inheritance. So if this is done, let me know. Have you noted this
No, sir.
example? Yes, please note down. So when Mendel did this, luckily he could not find any recombination in the genes that he studied. So that's why in science we say that, you know, any fundamental experiment or law or theory should be tested with proofs, and as much as we can test it through in different organisms. So whatever is universal will hold true in every organism. For example, we make proteins from the RNA and we make RNA from DNA. This is true in every organism, right?
Yes, sir.
Yes.
Sir, done.
Done, everyone?
Yes, sir.
So let's now go to chromosomal theory of inheritance. Now let me tell you a very interesting thing before that. So you know that all this work, all these laws that we study, the three laws, actually Mendel did all the work, but the way he wrote his work was like a book, like novels. So he kept noting down every observation of his, and he started telling that this is the ratio and this is where the ratios are different in the F2 generation and why are they different. He talked in factors. So there, he did not know about genes at that time. He's the father of genetics. So he started this inheritance and factors can be inherited thing. So no one knew anything about gene at that time. No one knew anything about that these are segments of the DNA that gets passed on in chromosome. So that's why chromosomal theory of inheritance came very late. And after Mendel did his work, just let me write down chromosomal theory of inheritance. after Mendel did his work for a very long time, for like decades, no one believed in Mendel's work. No biologists bought Mendel's work. So they thought that whatever Mendel did, it did not get popular. It was not widely accepted. There were few people who thought that, yeah, it makes sense. But most of the contemporary biologists of that time discarded the work. Now, there were three reasons behind Mendel's work staying unrecognized. And the status of laws was not at all there. So no, people were not recognizing his work only, let alone that something is a law or something. Okay. So there were three reasons. First is, as we have already discussed, right, that Mendel was not a biologist, right? We have discussed this.
Yes, sir.
He, for the first time, used mathematical concepts of ratios and crosses and recombinations, permutation combinations to explain biology, which was in itself a very unpopular and a very unique thing to do. So biologists did not accept it straightforward. Second reason was that he could not widely publicize his work. So in those days, publications, so anything in today's era of internet, the world is a very small place. You know, we all are sitting miles away from each other, thousands of miles away from each other, but still we are connected. We can discuss things and get educated, right? So anything that happens at your place, I can get to know about it as fast as internet allows, right? Yes or no? But in that time, it was not the case. So scientists used to meet, like in Europe, people used to work for years, and then they used to meet at one place after every year or every 3 years or 4 years, and used to discuss what they have figured out with proofs. And then everyone used to question on that. So it's like a conference. But Mendel could not reach out to many biologists, and his work could not get widely publicized because of lack of communication. So his work remained like in the book that he compiled, and it stayed there. Second reason I've told you, he used mathematics to explain biology, which was not normal and not widely appreciated. Third was, he could not explain what is getting inherited. So he kept saying that there are factors, there are discrete factors that go from one generation to another, and these factors do not blend. So because he proved it in pea plant, that either you have a factor for tallness or you have a factor for dwarfness, and they both do not mix. So because he did not know about incomplete dominance, he did not know about codominance, he did not know about linkage. So he kept saying that these factors do not blend and they remain discrete. And biologists did not buy it because if you look at human height as a character, do you think that humans are either tall or dwarf? These two categories?
No, sir.
No, right? You will also not see that there is an upper end and a lower end. There is height ranging from 4 foot to like 6-7 foot, and every inch you will get, right? A person with 5'3", 5'4", 5'5", 5'6", all the way up to whatever is the tallest human on the planet. So there is a whole rainbow spectrum. Yes or no?
Yes, sir.
Yes, sir.
So when he started talking about his work, scientists just discarded it, saying that it does not make sense. We have human of all varied heights, and we have humans of all varied skin color. Do you think that we are either white or black? No, right? We are of, sorry?
So I was just answering, sir.
Yes, please tell me. What were you saying?
Sir, I was saying that we are not of all. We are all not of same color.
Yeah, and we have a gradient, right? So if you know anything about the makeup or like the facial things done that is used for film industry and things, you will see that there is shade of the skin. For the first time when I went to one of these beauty salons, and I saw that there was one wall full of different shades of foundation for human skin color, ranging from the darkest skin to the fairest skin. And you will see that it's like the palette is like a spectrum. And it makes you think as a biologist that this is all different types of skin colors, which are possible because of different concentrations of melanin in the skin, right? So Mendel could not explain, if your genes are not blending in, then how is this happening? He was correct. Alleles were not blending, but because he could not explain this thing, people did not, bought his work. Scientists did not bought his work. So write down. Mendel published his work. So you have to know certain things. So Mendel's work got published in around, mid 19th century, which is around 1865, if I'm not wrong. Please confirm this year. 1865 or 55. Okay, so it got published here, and it was not accepted till the beginning of the 20th century, which is till 1900. It remained unrecognized.
Sir, it's 1865.
Sorry,
Sir, it is 1865.
you confirmed, yeah?
Yes, sir.
Okay, where did you confirm from? With the
Google.
source. Okay, please cross confirm also. I have a little trust issues with Wikipedia and Google these days, and it
was
remained
9 February 1865.
okay. Now I don't have issues. So it remained unrecognized till 1900. Like this I remember because 1900 is where 20th century starts. So the beginning of 20th century also saw the beginning of genetics. Like it was the field of genetics reviving, because in 1900 there were 3 scientists who independently figured out Mendel's work, and their names are, one was Hugo de Vries, second was von Schermach, and third is Carl Correns. So these 3 scientists independently rediscovered Mendel's work. They found that publication and they read it. Okay, this work. And when they read it, they realized that Mendel did something very, very logical and interesting. And for like 35 years, no one gave any kind of attention to it. And it was very, very, disappointing to them. So Hugo de Vries is the person who's mainly responsible for reading Mendel's work and raising the work to the status of laws. So he figured out, actually they all figure out, but Hugo de Vries, he figured out that all his work can be summarized into 3 laws. So the laws that we study, which is called law of Mendel, was not actually given by Mendel. You understand that?
Yes, sir.
Do you understand? And this is the ethics and ethos of science in that period. See, these scientists knew that Mendel have invested his life studying this. Now he cannot say that Vries laws of genetics. He still called it Mendel's laws of genetics because Mendel did the work. It was his science, his finding. He just redefined it in other language to just give these 3 specific laws. So the status of laws was given by these 3. Let's write down, not by Mendel. And also in the year 1900, another thing happened. There were two more people whose name was Sutton and Boveri. They also read Mendel's work, and they realized by this time, like it is 35 years later, by this time people knew about chromosomes. We know that chromosomes are the thread-like structures in cell which contains the genetic material, and it gets inherited from one cell to another when the cell divides. So Sutton and Boveri actually gave the chromosomal theory of inheritance. So write down. Sutton and Boveri gave chromosomal theory. It is this theory validated Mendel's work for the first time in biology, and people started realizing that the work Mendel did is, immense, and it holds immense reputation, and it's valid. So chromosomal theory of inheritance. And they explained what Mendel could not. That what are these factors you are talking about? So they said these factors are basically the genes on the chromosome. You understood?
Yes.
Is it clear, everyone?
Yes, sir.
Yes. So write down. Sutton and Boveri gave the chromosomal theory of inheritance, which states, this theory states that the pairing and separation, whether you are pairing or separating a pair of chromosomes, the pairing and separation of a pair of chromosomes would lead to the pairing and segregation. The pairing and segregation of a pair of factors. In bracket, write down alleles, because now you know what are they that they carry. Do you understand this statement now? So this validates Mendel's work, that basically the alleles that factors that Mendel talked about are alleles which are present on chromosomes. So if you pair two chromosomes, you are pairing the factors together. And if you are separating two chromosomes, you are separating the factors together. And this is how gametes are formed, right? One copy of the genome goes to one cell and the other copy goes to other cell. And that's why a primary spermatocyte, which is diploid, becomes two secondary spermatocytes. Yes, carrying one allele each of every gene. Do you understand? Can you now connect it to the reproduction unit? Everyone? Everyone, am I audible? People, am I audible? Yes or no? Fatima, Sumaira, Arpit?
Yes, sir.
Yes? Okay. You can also write, so basically Sutton and Boveri united the knowledge of chromosomal inheritance with the knowledge of Mendel's laws and just validated it. So remember who gave the status of laws? It's these three scientists. And who gave the chromosomal theory of inheritance? Sutton and Boveri. Okay.
Can you scroll up?
Yeah. Let me know when you're done. The next topic that we will do is the most crucial, confusing, if you do not understand it completely.
So done.
Done? Everyone?
Yes, sir.
Smaira, Fatima?
Yes.
Yes, sir.
Yes. So now let's come to, I've already told you about linkage. You understood linkage, right? So if linkage is there, there will be no recombination. But now let's try to understand what is recombination. This is the process by which sexually reproducing organisms produce variations. Right from chapter 2, you have been studying that sexual reproduction produces variations. Now you are going to know the reason, the actual mechanism, how it happens. Okay, because you know now understand the genetics. So recombination, write down. It is the production of non-parental type gene combinations. Through meiosis. Okay. Now, to understand recombination, you have to understand certain things like what are homologous chromosomes. You understand already what are chromosomes, right?
So
Do you understand what are homologous chromosomes? Anyone? If this part has been done in your school, do you understand what is a homologous chromosome? What are chromatids? And what are sister chromatids and non-sister chromatids? Are you clear about it? If anyone knows anything, please speak up. No problem. And then you also have to understand chromatids, sister chromatids, non-sister chromatids, and genes and alleles you already know. But you do not know crossing over. So these three things we will understand now. So pay attention, okay? And this is the most crucial part. One copy of the genome comes from mother. Yes, keep saying yes or no, so that
Yes,
I know
sir.
that you, yes. One copy of the genome comes from father. Sperm and ovum, right? They fuse together, make a zygote. So in a zygote, everything, every gene has two copies. Yes or no?
Yes, sir.
And how many chromosomes do human have?
46.
46. How many chromosome pairs do human have?
23.
23, which means 23 chromosome came from mother and the same 23 chromosome came from father. Yes or no?
Yes, sir.
So when they come like this, like a tiny thread-like structure. Okay. This is our chromosome. So let's say this chromosome, which I'm making here in blue, is chromosome number 1, for example. Okay. So this is chromosome number 1 from the mother. Okay.
Yes.
The same chromosome number 1 will also come from the father side. So I'm just using different color to make it, but you can understand, right?
Yes, sir.
This is just color coded differently, but it is the same chromosome, chromosome number 1. This is chromosome number 1, which came from father, correct?
Yes, sir.
Now these two chromosomes, because they basically are same, so they are called homologous chromosome. Homologous, the word homo and hetero you already know, right? Homo means same, homogeneous mixtures which cannot be
Yes, sir.
differentiated apart. Yes. So homologous chromosomes are basically the chromosomes which has everything same. The same number of genes, the same genes, but what they can have different is the different alleles for the same gene. You understand? So first, just know that these two chromosomes are, okay, let's not make it here, this part as well. I'm just removing this part so that we can use it better. So these two chromosomes are called homologous chromosomes. And write down what is the characteristic of homologous chromosomes. They have same length. Okay. And before that, let me tell you the basic features of one chromosome. So let's say this is one chromosome. Every chromosome has a short arm. Can you see this? So in the center, there is a centromere. Now it is not exactly in the center for most of the chromosomes. It can be slightly up or down. It can also be towards the very end. And centromere decides that there is a short arm. Can you see this is shorter? Yes or no? And then there is a long arm. Is it understood, people? In every
Yes, sir.
chromosome, the short arm is called the P arm. And the word P is for petite. You know what does the word petite means? Tiny or small. Petite, it's an English word. And the longer arm is called QR. Okay. And then there is a centromere. Here also, it's the centromere, the P arm, and the Q arm. Okay. So in a homologous chromosome, the total length, and this is the total length of the chromosome. So they have the same total length. They also have same length of P and Q arm. Okay. They have same number of genes. And all the genes are present at the same locus. So they have same locus of alleles. Now what are alleles? Let me just show you. For example, let's say there is a gene present here. You see this red mark? At
Yes,
this
sir.
place, there is a gene present for height, and the allele it carries is capital T. So this has a allele which is the dominant allele, and if this is inherited, it will give rise to a tall height. So at the very same locus on a homologous chromosome will be the same gene, but it is possible that this might carry a different allele. Do you understand? But the locus will be same. So this is known as locus of allele, or position, physical position where the gene is present on the chromosome. Is it clear, everyone?
Yes, sir.
Yes.
Yes, sir.
If anything is not clear, please tell me. We will discuss it multiple times. No problem with that. Now, let's say there is another gene, and that is very close. I think I should use this one. Can you see this yellow mark? Is it visible, people?
Yes, sir.
Yes. And let's see, this carries the gene for seed shape. So this carries capital R and this carries small r. Okay, for example. And let's say there is another gene which is present here on the other side. And this is for yellow and green seed color. So this has capital Y and this here will have small y. Is it clear?
Yes, sir.
Yes. So this and this, they are genes. 3 different genes present at different location on a chromosome. Their location is known as the locus for the gene. Okay. So you understood what is homologous chromosome? The chromosomes which are basically the same chromosomes coming from 2 different gametes. They contain the same length of the short arm and the long arm, same number of genes, and same location of all the alleles or the genes. Okay. Now, just before cell divides, so they come as, if they are single, they are called chromatid. If it is one, it is called chromatid, right? This chromosome is in the form of a chromatid. Just before cell division, this replicates. Remember, I told you that the DNA content replicates, right?
Yes, sir.
So this replicates. And every chromosome now has 2 chromatids. One chromatid replicated from the other. Is it clear, everyone? Say yes or no?
So,
Similarly, it will happen here. Okay. So there will be a replicated chromatid. Yes. This happens just during meiosis. Okay, now let's mark everything back. This is centromere and this is Y chromosome, Y gene, or I can write it.
sir,
Yes, tell me.
sir, the replicated chromosome will contain the same genes.
Yes, of course, because it's replicated, no? If you are making a photocopy of a page where you had your notes, the other page will have different notes.
No, it will be same.
Yeah,
Sir, it will be same.
it will be same, correct? So this is just a photocopy, because before the cell division, the chromosome needs to make 2 copies or not. It come as this, but when the cell divides mitotically, so in mitosis, sorry, meiotically, in meiosis 2 is just like mitosis, yes or no?
Yes, sir.
So this gets segregated, then it has to replicate itself so that it can again get segregated. Yes, meiosis
Yes,
2,
sir.
people.
Yes, sir.
Yeah, so it has to make its copy. Now when it makes its copy, then these 2 copies, these 2 chromatids, because they are exact copies of each other, they are called sister chromatids. What are they called? Sister chromatids. So sister chromatids are the chromatids of the same chromosome. And if I take this one and this one, they will be called non-sister sister chromatids. You understand?
Sir, can you repeat?
Yes, I will.
Sir, the sister chromatids will have the same genes, right?
They will have the same alleles also. See, Arpit, when a chromosome, when it got inherited, it was like a thin thread, one single thread. Then just before like getting segregated into two different cells, it got replicated, right? So
Yes,
one
sir.
thread became two. And these are the two threads, the green ones, two green one. So both greens, because they come from the same chromosome, yes, will be
Yes.
called sister chromatids. Sisters are born in the same house, right?
Yes, sir.
And these two will be sister chromatids then, right?
Yes, sir.
These two are also sister chromatids. Now, sister chromatids have exactly the same allele for the gene. Okay.
Okay, sir.
If recombination has not happened, so right now no recombination has happened, so the sister chromatids will have exactly the same alleles of the same genes present at the same location. So if I just explain you, here also there will be a small t, and here also there will be a capital T. You understand?
Yes, sir.
Yes. And here also there will be a small r, and here also on this sister chromatid there will be a capital R. You understand?
Yes, sir.
Yes, sir.
And similarly, here also there will be a capital Y, and here there will be a small y, right? So these two are called sister chromatids, the two greens and the two blues, and one blue and one green are called non-sister chromatids. Is it clear? But
Yes,
non-sister
sir.
chromatids belong to homologous chromosomes. Is it clear, everyone? They still are same chromosomes, but the chromatids of two individual chromosomes are called non-sister. One coming from like chromosome 1 from mother, chromosome 1 from father. Is it clear,
Yes, sir.
everyone?
Yes, sir.
Yes, sir.
Making some space. Please first write everything: centromere, locus, short arm, long arm, sister chromatid, non-sister, and do it very, very meticulously and carefully. Then I will tell you how recombination happens. Please use two different inks if you can, because in the next part you will be needing different inks. You all can still see the screen, right? Fatima, Arpit, and Samaira?
Yes, sir.
Yeah, okay. I'm just trying to. Let me know when done, people. You can take few more minutes, but make it clear, okay? So that when you yourself come back to it, you can understand everything clearly. Okay, finish and let me know. I'll just come in a minute. Done? Everyone? Arpit, Fatima, Sumaira, done?
Yes, sir.
Yes, sir.
Okay, very good. So now when you understood homologous chromosomes, sister, non-sister chromatids, and everything related to it, let me start with something known as crossing over. So this is how recombination happens. So basically what happens is, let's come back to the same two chromosomes. So this time I will make it like this. Can you see these? These are the same two chromosomes. And what genes did they carry?
Capital T, capital R, and capital Y.
Yes, but I'm asking what genes? You're correct, not wrong. The genes was for like height, seed shape, and seed color, right?
Yes.
But on this chromosome, you had what? Capital T, yes. And then you had capital R, correct? Let's say. And on the other arm, you had capital Y and capital Y, correct? Let's make another chromosome as well, the homologous chromosome. This is the homologous. And what were the alleles for the same gene present on homologous chromosome? I prefer to draw it in every class, so it's like a tedious task, but instead of just showing picture and going hastily over it, I've seen that this works. This whole process, if it gets into your head, you will never forget recombination. And that's my motive. So it will have, tell me.
Small t, small t.
Small t, and
Smaller, smaller.
small r, and
Small y, small y.
small y, and small y. Correct, everyone? Now, during a crossover, what happens is this state where these two are present like this in a cell, they start forming a chiasmata. So both the chromosomes come closer, okay? And they form a chiasmata. A chiasmata is a crossing over event in both these chromosomes. Do you understand now what's happening? The two chromosomes came close together and overlapped a part of each other over each other. Just like two chromosomes playing bhangra. Both the hands up in the air, one leg on the ground and the other crossed. You know how to dance a bhangra, right? Everyone?
Yes, sir.
Yes, Arpit is sometimes, like when he sees no one is active, he just wakes up the whole batch. Good, Arpit. Like your spirit. So these two chromosomes are here playing bhangra, crossing each other's leg. It can also happen anywhere. It's not important that they will play bhangra with the leg only. Sometimes they clap together. So it could be the short arm overlapping each other. It could be the short arm and a long arm together. But to not confuse you, I'm just using one location where recombination is happening. Now again, let's mark. Sorry, red it was. Capital T, small t, yes?
Yes, sir.
Capital R, small r, yes?
Yes, sir.
Capital Y, here it was capital Y, and somewhere here only small y and small y. So where the recombination happened, where this crossing over happened, so this stage is called exchange of genetic material between two chromosomes. Okay. And this, which I'm drawing here, this part is called a chiasmata. Chiasmata or crossover bridge. Is it clear?
Yes.
Okay. There are many names. Now, because of this, what happens basically is there will be exchange. So where they overlap, one part where they overlap, the part gets broken, and one part gets attached to the other chromosome, and the other part get attached to the first chromosome. So in recombination, it is physical breaking apart and joining back of the pieces of chromosome to non-sister chromatids of a homologous chromosome. Now these two, the chiasmata, is being formed between, what are these two, sister chromatids or non-sister chromatids?
Non-sister.
Yes. So that's where the concept of sister and non-sister is important. Because if you do crossover between sister chromatids, will that crossover be beneficial?
No.
No, they are the same copies. But if you do it between non-sister chromatids, what can happen is interesting. So what will happen now is, when they again will be separated, ready to go back in their respective cells,
So you can use a highlighter to color it. It will be. I
yes,
guess.
but the point is,
Yes, it has to.
yeah, you guess right. I can use a marker. Oh yes.
Yes, sir.
Thank you so much, Arpit. So wise of you, man. I don't know when I will become like you. Ah, where were you, my savior? I've been doing this for all the batches. Arpit's like, this man is really slow. He needs some advice. Thank you. But this will happen. You understand?
Yes, sir.
And similarly, there will be another chromosome, another, in which you will get this. You understand? Everyone understands?
Yes.
So let me go back to pen. So if I now, this is the centromere, if I now mark the genes, so this will be, oh, tell me, capital T,
Yes, sir.
small t, yes,
Yes, sir.
capital R, small r, yes. Now this is the magic of biology. Capital Y, small y, capital Y, small y. You see this?
Yes, sir.
So because of this exchange, the physical exchange, what happened is, now there are 4 chromatids here. These two are sister, and these two are non-sister, right? So let's say this is chromatid 1, this is chromatid 2, this is chromatid 3, and this is chromatid 4. So you see that chromatid 1 is still parental type, and chromatid 4 is also still parental type, right? So chromatid 1 contains capital T, capital R, and capital Y, and this contains small t, small r, and small y, right?
Yes,
And
sir.
both these are parental, right? Or we call it non-recombinants. Yes,
Yes,
there is no recombination in these two. Non-recombinants. Oh, no. Sorry. Arey yaar. What is happening? Why is this happening? Okay, good. Recombinants. Whereas if you see these two chromatids, the chromatid 2 and chromatid 3. So let me use another color. Chromatid 2 here and chromatid 3 here. Do you see some change here? It contains capital T, capital R, but small y. And this carries small t, small r, but capital Y. So these two are basically what? These two are recombinants, right? Yes or no?
Yes, sir.
So you see that two are recombinants and then two are non-recombinants. Yes? Or parental. Is this clear, everyone?
Yes, sir.
This is how in biology, when we say in sexual reproduction meiosis happens, this is how variation comes up. Variation happens. So this part was normal, then exchange of genetic material was happening, and in this part you see that we actually have a recombinants. Now this combination was not present in any of the parent. Yes, that's why they are called recombinants. Now imagine if it was happening at two places between T and R also, then we will get even more recombinants, right?
Yes, sir.
Yeah, so the more recombination happens, more recombinants you get. Here I am giving you an example of just recombination at one place in the chromatid to explain you, and it always happens between non-sister chromatids to give rise to variations. Now please draw this, note everything down and let me know, then I'll move forward. Is the event clear to all of you?
Yes, sir.
Is there any doubt, Samaira, Fatima,
Yes, sir.
Arpit? Fatima and Arpit?
Sir, clear.
Clear? Fatima?
Yes, sir.
Very good. Please draw it. Okay, done. Arpit, Samaira, you're done?
No, sir, not yet.
Yeah, take your time. But when you're making it, make it neat and clean, okay? Clear, so that you understand this.
So,
Yes,
can you explain this flow chart that you made on the last time? No, so
which
there
flow
on
chart?
some sort of flow. Except this recombinants wala.
This one, okay.
Yeah.
So there are 4 chromatids, right? 1, this 2. So this is the one chromatid. This is the other chromatid. Yes.
Yes, sir.
This is the 3rd chromatid, and this is the 4th chromatid. Out of 2 are sister, 2 are non-sisters. Right?
Yes, sir.
You see the first chromatid, which is this one. If I mark it, this first chromatid you see, it has capital T, capital R, and capital Y as a combination. Yes.
Sir, will it not be taken in cross
Like this?
Yeah.
It does not matter. No, because when they replicated, so when you divide it from half, you can divide it like this. You divide it from half. One is one and the other is other. Okay.
Yes, sir.
Yeah. It's not any rule. So capital T, capital R, capital Y, parent type, right?
Yes, sir.
These are the parent types. Then again, you see the 4th one, which is this one, small t, small r, small y, right? Parental. But you see these two, capital T, capital R, and small y. Can you see this one?
Yes, sir. Yes, sir.
And this one is small t, small r, and capital Y. Can you see this one?
Yes, sir.
So these two are recombinant chromatids, and these are parental type chromatids or non-recombinant chromatids. Okay.
Yes, sir.
So in these two, recombination has happened. Clear, Sumaira and Arpit? Shall I move forward?
Yes, sir.
Yeah. Okay. So let me. Do you all have NCERT with you?
Yes, sir.
If you have NCERT, you can go to page number. Put it here. Page number 84. And I'm trying to import that image here. Yes, can you all see this image?
Yes, sir.
This is from NCERT. This talks about linkage and recombination, parental type, things like that. Is this image visible to all of you?
Yes, sir.
Yeah, so let's analyze this image. Okay, so look here. This is cross A, first cross, and this is cross B. These are two different crosses. Now in this parental type is Y and W for yellow and white. Yellow body, white eyed. This is the exact cross we are discussing what Morgan did. And on the other hand, if it is yellow and white, this Y plus and W plus will be brown and red, right? Yellow, white, brown, red. Then in the F1 generation, and also pay attention, what is the distance between these two genes on a chromosome? So this is a chromosome. And this here is centromere. Do you see centromere at the very end? In a chromosome, if centromere is at the very end, then this is the P arm, and there is a very small, this is the P arm, and this is a Q arm, right? And this is the centromere, correct? And these type of chromosomes are called telomeric chromosome because the centromere is towards the tail end. Okay, so don't get confused by any of the notations. It's just like, so they are showing here chromosome in the mother. So this is the female and this is the male. And you see when same when F1 generation came up and when before the F2 generation, when recombination happens in meiosis, you can see this Y plus and W plus came together, right? And there is very less. How many are parental types? Look here, the ratio, the percentage. How many are parental types? 98.7% are parental type. And how many are recombinants? Only 1.3. So by looking at the parental and recombinant percentage, what can you tell about these two genes? The gene between yellow body and white eye, the Y and W gene. They are very close to each other on the chromosome. And above also I showed you genes which are close to each other, there are less chances that they will get separated. It's simple logic. Now look at my finger. You can see my finger here. So there are two lines already here, right? In this one, you can see this is not single. This is 2, 1, 2, 3, and 4, right? Forget about this. Just look at these 1, 2, and 3. Can you see these lines?
Yes.
Now let's say these are 3 genes, 1, 2, and 3. Now these 2 genes are very close to each other, right?
Yes.
Whereas this gene is separate from here. Now, if randomly, these crossing overs are random. Now, if randomly my finger has to be chopped off so that again it can rejoin like a chromosome. Let's say this is chromosome. And if randomly it's chopped off, then the chances of chopping off is anywhere, right?
Yeah.
But in most of the times, the chances between this gene and these 2 genes, in between there is a lot of space. So chances of these 2 genes, A and B, getting separated is more, right?
Yes.
Because there is distance between the 2. So less linkage, more recombination. But what are the chances of this B and C getting separated?
Very less.
Very, very less, because it has to be very precisely, the blade has to run through this between a very narrow region between these 2 genes, right? And if it hits here, then only these 2 genes can get separated. You understand? Yes, everyone?
Yes.
Similarly, on a chromosome, if 2 genes like R and Y, look at this, R and Y, they are very far from each other. If you randomly make a chiasmata and cut that part, you can random make cuts anywhere, right? Any of these cut will independently segregate R and Y, right?
Yes.
You understand? But in order to, sorry, in order to, how can I get rid of this? Let's forget. In order to get T and R separated, we have only very few limited options. Either the cut has to be here or here, right?
Yes, sir.
Do you understand, everyone? So there is a physical thing that working between them. The physical association, the physical closeness, proximity of 2 genes on a chromosome leads to their linkage. Closer the genes are on a chromosome, if 2 genes are adjacent to each other, like they are just neighbors, then the chances of these 2 genes getting separated is very, 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 very less. There can be 98 or 99% linkage between the 2. So parental types, look here, this parental types denote linkage. What does it denote? And only 1.3% are recombinants because they are 98.7% linked genes. Do you now understand why did Mendel received a phenotypic ratio which was different from 9:3:3:1? Do you understand? Fatima and Sumaira?
Yes, sir.
So is the first case clear to you where parental is more and recombinants is less? Now let's come to cross B. They have taken two genes again. Now this time it is for white eye, but miniature wings, like the size of the wing. Now these two genes are located far from each other. Can you see the distance between these two and these two? They are
Yeah.
very close, they are very far. Now if they are far on the chromosome, you will say the parental is 62.8 and the recombinant is 37.2. What was the percent of recombinants in the dihybrid cross F2 generation? I asked you to find percentage. Someone found the percentage and told me. What was the percentage? Let's go up. How much was it?
37.
So 37% of recombinants means following the rule of Mendel. Yes? Now, how many recombinants do you see here?
37 point.
So the phenotypic ratio will be 9:3:3:1, right? It is following the law of Mendel, yes?
Yes.
So this is the normal law. Less linkage, more recombination. Is it clear, everyone?
Yes, sir.
Okay. Very good. So linkage and recombination both is clear to you. Oh, pen. So now let's start with two more concepts which are important for entrance and boards both. Till now, whatever we have studied was according to Mendel's work, according to Mendel's law. So Mendel gave certain laws, and those were the three laws. So we studied the three laws and we studied the exceptions for the two laws. Law of segregation is pure law. Now there's something called polygenic inheritance. Anyone knows what is a polygenic inheritance? If this part is done in your school, you must have studied this part. Fatima, did you do polygenic inheritance in school?
So I don't remember.
Okay. So remember in the very beginning of the today's class, we talked about human skin color and human height as characters, right? Yes or no? What was unique about both these characters? That human height has a spectrum. It does not fit in like tall and short, and human skin color does not fit in black or white, dark or fair, right? It has a spectrum. So these kind of characters are the characters which are not controlled by one gene. They are controlled by multiple genes. So let's define it. Write down. Characters or traits that are controlled by now this is something different from Mendel. Mendel did not know about this thing. Characters or traits that are controlled by more than two genes. More than two genes are called polygenic characters. Are polygenic traits or characters. And next point, write down. Polygenic inheritance is also regulated by environment, like environmental factors. Is it clear? Example is the best is human height and human skin color. Now let's take the example of human skin color. We know that human skin color is like affected by the genetics, the genes, and is also affected by how much sunlight it is exposed to. Yes or no? We get tanned when we are in sunlight.
Yeah.
Fatima and Samara. So, which means that it's not only the genes which can upregulate or downregulate the expression of melanin, but it can also be physical factors, right? Like exposure to sunlight. So in human skin, write down. Human skin is affected both by the genes and the environment. In bracket, exposure to sunlight. Now let's assume that human skin color is controlled by 4 genes. Okay. A, B, C. You can assume any number of genes. I'm assuming 4 genes. It has to be more than 2. So let's assume there are 4 genes. Okay. Genes regulating skin color. In this case, so if A gene, B gene, C gene, and D gene, all these genes are responsible for somehow formation of melanin in the cell. Do you understand, everyone? Samira and Fatima, do you understand?
Yes,
So
sir.
melanin is formed when these genes are expressed. So if both these genes, all the 4 genes are present in the dominant homozygous condition, A, B, B, C, C, and D, D. These are the 2 alleles. Now this combination where all these 4 genes are expressing melanin, this will be the darkest skin color. Am I correct?
I didn't catch that.
Siri gets activated without any reason. So this is the darkest skin color. You understand? And what will be the lightest skin color?
Small a, small a, small b, small b, small c, small c, small d, small d.
Yes, when every of these gene is recessive and not producing melanin enough to give skin a darker shade. So this will be the fairest skin color, right? In between these two, you see there is a gradation, right? If a lesser tone than this will be when only one gene becomes homozygous recessive, like this. So this will be less darker,
Sir,
right?
sir,
Yes. But
how many genes control for skin color?
see, till now, as far as I know, there can be more than 6 genes that actually code for skin color in humans, but those genes are still under research. Like all point is how do they interact with each other? For height also, there are multiple genes that affect height. And all these genes, do we get from the parents in active state or inactive state, or recessive or homozygous or dominant homozygous? It depends on that also. So we do not know for sure. We know till now there are around 6, but there can be more as well.
Thank you.
Okay. Yeah. So I am assuming here, just to show you the combination, that this will be the darkest skin color and this will be the fairest skin color. And this is why, because this is multigenic inheritance, this does not disprove Mendel's work. It just tells that this is something that is not following Mendel's rule. And because this Mendel could not explain in his time, his work was not appreciated. But now we know, still it holds true that there are 4 different genes affecting one phenotype, which is called polygenic trait or polygenic phenotype. And height in pea plant is not polygenic, but height in humans is polygenic trait. You understand? That's why Mendel could not explain it. And as we keep going down, like let's say there are two of them which are dominant and the other two which are recessive, this will be even lighter, and similarly we can make multiple permutation combinations, right?
Sir, see, there are so many different colors in human skin. So in this particular example, like only a few of them are possible. Like as
Yes, because we have assumed only 4 genes, right? When you assume
but
6 genes,
even if it's 6, then it can be only like it can be only like capital C, capital C, small c. What will it show? What will be the character? Means like what will be the color?
you're saying this one, right?
Yeah, along with all of them capital.
Yeah, this will also be a lesser shade than, because here also one is recessive, one is dominant, and we do not know the interaction between them. So one allele is not producing protein, so that will give another fairer shade. And if there are 6, then 6 raised to power 6 will be the total permutation combinations. You understand?
Yeah.
So 2 raised to power 6 basically. If you do this, what is this number? 2 into 2, 4 into 2, 8 into 2 into 3, 4, 5, 6, 4, 8, 16, 32, 64. So 64 different shades are just possible if we go through heterozygous and homozygous permutation combinations. And all these 4 genes are not equal in strength. Maybe C is the most strongest of all the genes. C codes for lot of melanin, and D does not produces that much of melanin. So if C is homozygous versus D is homozygous will also make difference. You understand?
Yeah.
So it's not like all the genes are equal in strength also. So even if they're equal in strength and there are 6, then we can have 64 different human shades. This is true, right?
Yes.
There can be 64 different human shades. So you understand, Fatima? Good question.
Yeah.
Okay. So write this thing down about polygenic inheritance. And I'll tell you one last thing in today written polygenic inheritance. So this is the genetics of it, and also environmental thing is more exposure to sunlight, then darker the skin, right? So these two, genetics and environment, together control polygenic inheritance. Same is for height. You will see that everyone gets a height, like the gene is decided for height, but if you still work out or have some activities during the childhood that helps in your bone elongation, like you must have heard that if someone is very keen towards gymnastics or towards climbing or hanging from somewhere or playing basketball, things like that, they help in your height shoot up. Yes, along with your genetics. Yes or no?
Yes, sir.
Same is for weight as well. See, you can get obese because of your genetics, but if you maintain a good activity, if you do gymming, you play sport, you exercise, then you can control it, right? Okay, so done. This is done.
Yeah.
Yeah. So polygenic inheritance is where more than 2 genes control only one character. Now just the opposite of it is pleiotropic inheritance, or also known as pleiotropy. Now write down what is pleiotropy. This is where a single gene single gene is responsible for multiple phenotypic expression or multiple characters. Do you understand? This also Mendel did not know. Many genes controlling one character is polygenic inheritance. One gene controlling many characters is pleiotropic inheritance. Is it clear?
Yes, sir.
And such a gene is called Pleiotropy gene. And one example of this, and it's a very, very famous example.
Phenylketonuria.
Yes, there is a disease called Phenylketonuria. So write down. Why is it not working? Let's save. Okay, it's working. So there is a disease known as Phenylketonuria. Now, in this disease, what happens is there is a single gene that gets mutated. Okay, so write down. A single gene gets mutated. Now, if it is a single gene, we will expect that it will just affect one character, just like gene for tallness was affecting height in pea plant, right? And this single gene basically is the gene that codes for an enzyme. And that enzyme is called, what was the name of the enzyme?
Phenylalanine.
Yes, thank you. Phenylalanine hydroxylase. So this is the enzyme for which this gene codes. And when it gets mutated, so if this gene gets mutated, you will think that one enzyme will not be formed and the character for which it will be responsible will not show up properly. But instead, we see that in this disease, in phenylketonuria, what are the symptoms? So, or manifestations, write down. Manifestations of phenylketonuria. Phenylketonuria affects, first is there is mental retardation, first symptom. So you're like, okay, fine. Mental retardation means what it is affecting? The nervous system, right?
Yes, sir.
Which means maybe this enzyme is for nervous system or for the brain. But it also affects depigmentation or reduced pigmentation of skin. Now, reduced pigmentation of skin. This is not nervous system, right? Yes or no? This is skin.
Yeah.
It also does reduced pigmentation of hair. Now, hair, skin, and brain. These are three different systems, right? So the effect of mutation in this one gene is multiple, right? There are multiple effects, multiple systems affected, right? Yes or no?
Yeah.
This means that this single gene, which codes for phenylalanine hydroxylase, controls many characters, because if this gene goes problematic, then you will see problem with nervous system, you will see problem with skin pigmentation, you will also see problem with hair pigmentation. Is it clear?
Yes, sir.
So that's why it is known as pleiotropic gene, and this phenomena is known as pleiotropic inheritance. Any doubt, Fatima and Samaira?
No, sir.
No? Okay, cool.
Sir,
So,
we didn't,
yes, Fatima.
we didn't
Yes, Fatima.
learn what is phenylketonuria.
We will do phenylketonuria in
Next chapter.
genetic disorders. It's a genetic disorder, but we have to, when I will teach you, no, in this chapter only, I will teach you pedigree analysis.
Yeah.
There we will do phenylketonuria. Okay.
Yeah.
Yep. This, I just use this disease as an example here to tell you that what is pleiotropic inheritance.
For us, teacher like completely explained it in detail by explaining Pleiotropy.
Yes, so we will do phenylketonuria in genetic disorders. There I will explain in detail and you will understand why it is pleiotropic.
Okay.
Okay.
Yeah.
Any doubts?
No.
First step first. Okay, then see you in the next class. Tomorrow I will be covering some major important portions for entrance, which is sex determination in other organisms like honeybees. A lot of questions come from honeybees and birds, cockroach, and I'll also do pedigree analysis and genetic disorders. So please don't miss the class. Okay. See you tomorrow. Bye bye.
Thank you, sir.
So, thank you.
Bye bye. Okay. Good evening, Aisha. Am I audible and visible to you?
Yes,
Yes. How are you?
I'm good. How are you?
I am very well. So, Aisha, let's start with some basic introduction. I hope you can hear me clearly.
Yes.
Yes. So, my name is Rahul, and I am faculty of biology at Learnivio, one of the faculties of biology. About me, my background is from biological research, and I'm pursuing a career in biological research. Apart from that, it's my passion to teach. I like teaching and preparing students for medical entrances, for research in biology, and for all other entrances where they want to pursue a career in biology. So tell me about yourself. How was your 10th class, and what is your motivation behind taking biology in 11th? What do you want to do? Just some basics.
My name is Aisha and I'm from 11th grade. I live in Qatar and I study in MS Indian school. And the reason behind taking biology is I like bio and I'm thinking of going into some medical fields in Qatar. So I
So you
don't
want
have
to
any. I'm not taking any NEET exam or anything. I'll be studying in Qatar itself.
okay. So, but you want to go into the profession of medicine. You want to become a
Yes.
doctor, or at least give a try to becoming a doctor, right?
Yes, yes.
You have not decided exactly what you want to do after your 12th, but you want to do something with biology. Like that's your
Exactly,
intention,
exactly.
right?
Yes.
Okay, brilliant. So, I think most of us, including me myself, started studying biology with the same intention, which is a good intention to begin with. Now, so welcome to Learnivio, Aisha, and I'll just share a screen with you. Tell me if you can see the screen that I'm sharing with you. So I've been
Yes.
told this is your trial class for biology, and in the trial class, I'll just also tell me a few things. Your classes for 11th has been started in school, biology classes.
Yes, it has started, and we have started with cell, the unit of life. We have started that chapter, and
Okay,
we have
and you
finished it.
started with
So yeah,
you
we
have finished cell. Okay, what are you currently doing?
Currently, we are doing anatomy of flowering plants.
Okay.
We have also
Currently on.
finished morphology, and we have finished cell. One more cell chapter.
Have you finished tissues?
No. We have finished cell chapter and cell unit of life, and one more cell chapter is there. I'm not getting the chapter name.
One is cell, which is the fundamental unit. One is cell division.
Yeah, cell division. Yeah.
So you have finished cell division as well.
Yes.
Okay. And you are currently doing anatomy because you have finished morphology of flowering plants. So you are on anatomy of flowering plants, right?
Yes.
So flowering plants are called angiosperms. Okay, cool. So cell, the fundamental unit of life, where you have studied about the organelles and all those things, and morphology of flowering plant and cell division. So the course in Learnivio you will be joining, it will start. So it will be a fresh batch, a new course. And I can do two things because I ask this to students to optimize my way of teaching. Now, let me tell you a little bit about how I teach. So basically, one thing that I have realized after like 6 years of teaching experience now, both offline and online, and including my own experience in the field of biology, is that biology has immense potential of becoming boring very easily. Correct? It is
Very
interesting.
true.
It is interesting a subject till I'm telling you, this is how your liver works and this is how it produces glucose and makes new glucose. You're like, oh, okay, very good. The moment I start dictating you things and telling you terminologies and definitions, you're like, I am out, I want to sleep. So that's the potential of biology as a subject. And I have realized the most difficult thing in biology is to memorize. There are a lot of things to memorize in biology, right? Especially the terminologies. Yes or no?
Yes.
And they are not normal English. they are biological terms. For example, cytoplasm. Very common biological term, but we never, because we never use this term in our daily life. You never go out and talk to your friends like, okay, you know, I touched a cytoplasm today. But we all in biology know what is cytoplasm. Similarly, there is another word called cytokinesis, right? Another terminology in biology. All these terms can come as one marker definition. In fact, you take up any chapter, you take any biological word and you can make it a one marker definition question. Define cytoplasm, define cytokinesis, define karyokinesis. And this is why biology becomes very tedious, because the students do not realize, and they do not know how much can you memorize. So this part I have started working on from last couple of years, that the memorizing part has to go down if you want to make biology excuse me, if you want to make biology great again and popular among students, decrease the memorization part. And this I do through a terminology section. So in my teaching, for both for class 11th and 12th, I suggest you have to make a copy, and the first few pages or the last few pages of your copy will be empty, where we will do this. You will write term and the meaning, like biological terminology and the normal meaning of it. So by the end of your session, like 11th class, you will have a mini dictionary of your own, which you have created. So you do not have to remember each and every word, because you know which word means what. For example, cytoplasm and cytokinesis, the word cyto in both. Do you know what does the word cyto means? It means cell. The chapter that you just studied. And you know what is the study of cell known as? Cytology. What is the study of life known as? Biology. Bio means life. Logy means study. Cytology means study of cell. Cytoplasm is, plasm means fluid. So the fluid content of the cell. The word kinesis means division. So if you know these two words, you will not have to remember the definition for cytokinesis anymore. You know it is cell division. The process of division of one cell to form two daughter cells is cytokinesis. You understand?
Yes.
Aisha? The same cyto word holds true here, 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 and 30 other places wherever it will come. If I ask you what is a cardiocyte, now a normal biology student will go on memorizing. But you know cardio? Have you ever been to any exercise session, or like if you like to run, they say that you are doing cardio when you are running. So what does cardio means? Go to a cardio specialist, cardiac surgery.
Something related to heart.
Perfect, brilliant. Cardio means heart. When we are doing cardiac exercises like cardio running, we are actually making our heart exercise. Okay, so when you are actually lifting weight, that is not cardio, that is your muscle, that is for your muscles to gain strength in that particular muscle. But when you are running, heart is starting to beat more than it should have. So that is called cardio, because it's the exercise for the heart. And you already know now what is a cyte or a cyto. What is this?
Cell.
Yes, so what is a cardiocyte? A heart cell.
Yes.
A cell which is, yes, a cell which is present in the heart is called cardiocyte. You just have to know that cardio means heart and cyte means cells. Okay, is it clear?
Yes, very clear.
So this is how I have started in my classes, reducing the memorization part of biology and making it more discursive and more interesting, because the more we focus on processes. So in biology, Aisha, now you are in 11th, so you have to change the way you read and study biology. Okay, till 10th, I could have given you for every chapter 10 important questions. So if there are 22 chapters in total, you have 200 important questions. And if you can memorize those 200 important questions, good, you score 80. Biology is a subject where passing is the easiest. Scoring 60 plus is easy. Scoring 80 plus is very, very difficult. This is how it works. Because to score 80 plus like in 90s or your 100s, you actually need to have two things. The zeal for biology, you have to be very, very motivated for biology. And second, less memorization, more understanding of the subject. So now in 11th and 12th, if you want to become a doctor, you cannot become a doctor by not understanding two things. In biology, there are two types of questions. What and how? What is this? What is that? What is this? What is that? And how this happens, how photosynthesis happens, how muscle contracts, like these. You understand? So what type of questions are based on your terms? Okay. What is cytokinesis? What is cytoplasm? What is the cardiocyte? How is based on processes. And for this, I do discussions in class. So whenever I start a topic, my classes are never going to be monologue that you are sitting in front of a screen. It's not a YouTube from where you are learning. The role of a teacher is that you can discuss with that person. You get a personalized classroom experience. So you can discuss, argue, you can give your logics to your answer, you can defend your answer, and then we can figure out what is the most logical answer. Is it clear, Aisha? This way, I try to prevent biology from becoming boring in my classes. Okay? You like it?
Yes.
Okay, good.
I love it so far.
Okay, very good. That's amazing then. I think you will be a very good addition to my. So all the students that I have taught till now, after, so they like biology. All of us like biology when we take biology, everyone. But the point is, they cannot endure biology. They cannot go through all that boredom and then emerge victorious at the end, because they always get trapped in terms, definitions, this and that. But if I tell you that, see, this is the beauty of this process, and biology is something which is happening inside you, right? You are a biological system, you're not a machine. So if, let's say, all the concepts of physics you cannot experience, for example, if I'm talking about a black hole, you are not in a black hole, right? Yes. If you are motivated, you will read that, understand that, be very happy about it. Same is with other subject, but everything that is happening in biology, from a cell division to synthesis of a protein, to breakdown of a protein, to any organ failure, any disease, any disorder, any genetic manipulation, is happening right inside you at the moment. Yes. So that's why it's interesting a subject. So I try my level best to keep it interesting, to do justice with this subject. Now, coming to your course, I can see that your school is not strictly following the NCERT, what do you say? The way NCERT has planned the syllabus from one chapter first to, and I also do not follow NCERT by heart. So I also start with cell. Okay. So when you will join classes, will it be okay for you? So when are your exams coming up, the next exam? Because we have to optimize two things. Yes. When is your next exam?
Our vacation is from 1st July to 15th August, and after that maybe in September. 1st of
Okay. So maybe September will be your exam. It can be early September also, right? So we have July and August. Okay, cool.
Yes, yes.
So yes. So let's do one thing. We will start from cell as the fundamental unit. Okay. It will be, if you have studied it well in your school, it will be a add up revision, and we will be able to finish it faster, and you will be in a small batch. So at Learnivio, we make small batches where every student can personally discuss things, and I can give time to each student and can know about their progress and can keep a track of their progress. So I will start with cell. and then I'll go to cell division. So these two things are very fundamental to biology. That's why most of the schools, and also I also believe that they should be taken first. Okay. So this will be the same from where I'll start. Then I'll go to morphology of flowering plants and we'll come to anatomy in the same way. Will it work for you? I do not want to leave something. So normally it depends on students. So if you want that, okay, leave these two things because I've already studied and start from here because I'm currently here, then I can optimize the syllabus like that also. But if you take my suggestion, let's start from cell. Because
Yes, I would want to start from cell.
yes, because I always believe that, you know, in schools, we do not cover everything, which is important for entrances. And that's why I believe you are making an extra effort to come out of your school education and pay somewhere else. And I do respect that, your efforts and both your finances that you are putting in here. And I will try to do justice with that. Okay, Aisha?
Yes.
Cool. So as you have already studied about cell and you have understood the way I teach, so terminology section is clear to you, discussion part is clear to you, and I see that you have no problem, you know, expressing yourself. You are very vocal, and that is one good thing. I like students in my class who are vocal and like to discuss. Now you have studied cell already, right? So let me give you some key concepts that you can take in like next 5 minutes. If you join my classes from next class onwards, I will start from here. So I want you to go. You can also take it as a homework. No, I'm giving a homework right in your trial class, but yes. So you have to just revise the cell part. And there is one thing, if you have not done in school, it's okay. There is something called central dogma of biology. Did anyone tell you about this? Central dogma of biology. All of living system depends on it. No? So you have studied that in a cell we have nucleus, right, Aisha?
Yes.
Inside the nucleus, what sits inside the nucleus? Why is nucleus so protected? It's called the brain of the cell, right? Why it is called the brain of the cell? I call it the safe vault of the cell. We are protecting the most important thing of the cell in the nucleus, which is our DNA. You know, DNA?
Yes.
And what is DNA? In normal terms, DNA is the blueprint of life. So whatever you look like, you behave like, your eating habits are, your likes, dislikes, your allergies, your diseases, you like certain kind of smells, you do not like certain kind of smells, same goes for taste, your favorite food. All of that, whatever makes you, you is encoded here in the DNA. That's why it's called the blueprint of life. Okay. Now this DNA is like a very long book containing every character of yours, but we cannot keep using. So if there is a book which you have to preserve for, let's say, 70 years, there is a code that you have to preserve for 70 years because you are going to live for 70 years. So would you like to daily open that book, like turn around the pages, read it, and then keep it, then again next day open it, turn around the pages? If you keep doing it, within 5-10 years, the book will be, you know, get old, right? Wear and tear will start happening. Yes or no?
Yes.
So we never access our DNA directly. You know, this is our blueprint, the original. So we always make, at one point of time, let's say your body wants to make a protein which is responsible for fighting against a disease. Suppose you were very happy, normal, and suddenly you got some infection. Now you have to make certain proteins that will help you fight that infection. And the recipe for that thing is in your DNA. Now you do not want to access the DNA directly, bring the DNA out of nucleus and access it in the cell. No. So instead, the DNA makes a messenger protein called mRNA or RNA. You know, RNA? DNA is double stranded, RNA is single stranded. This you know, right? From class 10th?
Yes.
So DNA makes a RNA mRNA is messenger RNA. It has the same message that DNA says. For example, let me give you an analogy. Who runs a country? The supreme leader. It can be the president or the prime minister. Yes. Who runs the country? In earlier times, it was used to be the king. Yes. So do you think that every decision was taken by the king? Yes, right. But does the king used to come out from the palace after every decision, stand on the choraha and tell what was the decision? No, right? He will send some messengers. Or same is in the case of a government. The president has certain ministries. So if the decision is about Ministry of Health, the health minister will come out and tell what is the decision, right? What are the laws? You understand? So similarly, DNA makes mRNA, and these mRNA can be converted into proteins in the cell. And since we have already studied cell now, you know that what are the protein factories of the cell? Who make proteins? From RNA to protein, there are structures called ribosomes. Remember? In your cell, these are organelles. Aisha? So these ribosomes synthesize proteins, and then these proteins play the functional role in our body. So everything is done by protein, but that protein comes from mRNA, and then mRNA comes from DNA. Now remember, the process of formation of mRNA from DNA is called transcription. It's just like you are making a script from the original source. So you have a book, which is the only copy. You do not want to degrade it. So whenever you want to say something, see something and take reference from that copy, you open the page, note down your own notes, close the book, keep it safe. You do not carry the book with you. So that's called transcription. We make transcripts. You understand? Now this mRNA is a transcript. Whether it degrades, whether it's destroyed, whether it is non-functional, we can make more transcripts, not a problem. DNA should not get degraded or destroyed. So this is called transcription. And from mRNA to protein, this process is called translation. Okay? Do you understand?
Yes.
This from DNA to protein, this is called the central dogma of biology. Every cell, every organism functions on this. So when I will be teaching you cell, I will also be preparing you for entrances and the kind of questions that any entrance, whether you give it in India, the NEET, or in Qatar also, to go into medical colleges, you have to sit for some exams, right? Or if it is on merit basis, you have to score good in boards. Yes? And for that, you need your concepts really, really strong. So this is how we will proceed. So your homework is you have to go back, revise cell, and also all the cell organelles. And when you are doing this, also revise what is the difference between a prokaryotic cell and a eukaryotic cell. That's all. This is where I begin from, the fundamentals, the very basic. So you understood the central dogma? Would you like to just note this thing down? It will help you, the homework in the central dogma. And if you understand the concept, if you like this, if you like this plan, then you are good to go, good to join. And I hope to see you in classes then. Okay, Aisha? What are your preferred days to study biology? When can you join biology? What days? Any preference you have? Because that I will convey to the coordination team when they will be making batch for you. Do you have any preferred days to study biology?
No, any day. I can do it.
Weekends will work. Weekends, weekdays will work, right?
Actually,
It
I
will
have
be-
other classes also. So.
Yeah, they will be coordinated according. Okay, if you have other classes also, so they will coordinate with you and will let you know, and will also let me know. Okay, so hopefully I'll see you in class somewhere this week only. Okay, Aisha, read this before you come to the class and other things we will discuss there. Do you have any questions for me, Aisha?
Yes, what is
Anything?
mRNA?
mRNA, yes. mRNA is the word M here is for messenger. You know, there are many types of RNAs in the cell. This our teachers don't tell us when they are teaching us cells, but they should. There is something called mRNA. This is the messenger mRNA. This has the message for the protein. There is something called tRNA. This is called transfer RNA. And this helps in the protein formation. And then there is another mRNA, which is mRNA, tRNA, and cRNA, snRNAs. So all these different type of RNAs are there. So that's why I used the word M. Is it clear?
Yes.
Yeah.
What is RNA?
RNA is, so DNA, you know, deoxyribonucleic acid, our genetic
I know.
material, a long thread. RNA is ribonucleic acid. So it does not have this deoxy component. This part is not there. And DNA is double stranded, RNA is single stranded. Just a single strand. Okay, that's the basic difference to begin with. Okay, Aisha, so I'll see you in the classes. Okay.
Yes.
Bye-bye.
Thank you.